It's a pleasure to be today with you. I'm going to talk about developmental origins of pediatric obesity, trying to focus on what I think we know from our work and that of others, primarily from our work, and highlight some areas where we could learn more to do more. So the outline of the presentation today is here. I'm going to talk a little bit about the burden of obesity and type 2 diabetes in youth, just to set the stage about why we're studying these conditions. Then move into developmental origins of these conditions and end with some thoughts about prevention throughout the life course. So, burden of obesity and type 2 diabetes in youth. We all know that the prevalence of pediatric obesity, childhood obesity, has been rising in this country and worldwide over the last 40 to 50 years. It has been rising steadily, and it has been rising both in adolescents and in young children, and even in infants. And after such a long time of a rising prevalence of obesity, it's perhaps no surprise that we are starting to see the chronic consequences associated with obesity in youth, namely shown here, type 2 diabetes, youth onset type 2 diabetes. Now, you may remember that we used to call type 2 diabetes adult onset diabetes, but now we're seeing it in children. And not only are we seeing it in children, we are seeing it rising steadily and rapidly. Here are data from the Search for Diabetes in Youth study, a surveillance effort trying to determine rates of pediatric diabetes in United States youth by age, sex, race, and ethnicity, and diabetes type. The Search study is showing us that type 2 diabetes in children has been rising steadily since 2003 at a rate of about 4.8% per year, shown here in the middle in red, but with higher incidence rates and higher trends over time in several racial ethnic groups, such as American Indians, shown here in light blue, African Americans, shown here in darker blue, and Asian Pacific Islanders, with the lowest rates and lowest trends, actually non-significant trends, in non-Hispanic white children. The type 2 diabetes exists in youth of all racial ethnic groups in this country and is rising rapidly. Based on the data from the Search study, our colleagues from the CDC have developed projections of the burden of diabetes, type 2 diabetes in youth over the next 40 years. So based on observed incidence data from 2010, they're projecting, if the rates continue to exist and the trends continue to exist as observed by the Search study, we're going to see the number of US youth with type 2 diabetes increasing by almost fourfold by 2050, from 22,000 youth to almost 85,000 youth. The highest increase is projected to occur among Hispanic youths, based on a combination of trends in rates of type 2 diabetes in this demographic group, as well as other demographic changes projected. So US healthcare systems need to be prepared. So with the rise in childhood obesity and now the rise in chronic conditions associated with childhood obesity and a younger age at onset of these conditions, no wonder that we're starting to focus our attention to risk factors that happen very early in the development that predispose these children for these chronic conditions early in life, the developmental causes of pediatric obesity and type 2 diabetes. So research in this field is following the framework put together by the Developmental Origins of Health and Disease hypotheses or framework. And this framework starts with a variety of exposures happening during sensitive periods of development, such as pregnancy and early life, shown here. So exposure during these sensitive periods to a variety of factors, where they be maternal condition, toxicant exposures, nutrition, stress, smoking, pollution, social environment during these sensitive periods lead over time to health and disease in individuals and populations. And these sensitive periods exist throughout the life course, outside of pregnancy in early childhood, mid-childhood. Puberty is another critical sensitive, developmentally sensitive period. And then again in pregnancy, thus perpetuating a cycle of virtual cycle, you can call it, or a vicious cycle of health and disease at the level of populations and even transgenerationally. So we've studied, according to this paradigm, a lot of environmental exposures happening early in life, and a lot of work has happened in the field of pregnancy diabetes. Exposure to diabetes during the intrauterine life was something that I and others have studied a lot as one of these potentially programming exposures. And these are data from two studies conducted almost, well, actually 10 years ago, 10 years apart. On the left side, among Pima Indians of Arizona, the population with the highest rates of type 2 diabetes in adults and in children. And on the right side of the slide, a study in a lower risk population, nested case control study within the larger SEARCH study, it's called SEARCH case control. So these two studies show that exposure to diabetes in utero is the strongest single risk factor for type 2 diabetes in youth. On the left, among the Pima Indians, what you see here is that the prevalence of the percent with type 2 diabetes is much higher in children who were exposed to diabetes in utero in blue compared to those who were not exposed in yellow, for an odds ratio of 10 for this association, a very strong odds ratio in epidemiology. And on the left, or sorry, on the right, in the SEARCH case control study, you see a much higher proportion of being exposed to maternal diabetes in utero among youth with type 2 diabetes in blue and among controls in yellow, for an odds ratio adjusted for age, sex, and race of 7.3, very close to the odds ratio seen in PIMAs. And note that the population in the SEARCH case control study is more diverse, include whites, Hispanics, and Blacks, yet the association between exposure to diabetes in utero and type 2 diabetes in youth is as strong as it is in a high-risk population, the Pima Indian population. Now, of course, you might say that if the mother has type 2 diabetes and the child has type 2 diabetes, the explanation must be genetic. Of course, that the offspring inherits susceptibility genes from their mothers. So in the Pima Indian population, we were able to determine whether that was the only explanation for the strong association that you just saw on the previous slide. So among the Pima Indians, we were able to select pairs of siblings discordant for diabetes and discordant for exposure for diabetes in utero because they were born before or after their mothers or fathers were diagnosed with diabetes. And on the left side of the slides, you see that among 28 SIB pairs discordant for diabetes, in 21 of the 28 pairs of siblings, diabetes was diagnosed in the sibling that was born after mother's diagnosis of diabetes. And only in 7 of the siblings, diabetes was diagnosed in the sibling born before mother's diagnosis of diabetes. For an odds ratio for the association between exposure to diabetes in utero or being born before or after mother's diagnosis of diabetes and type 2 diabetes in the offspring at a young age, an odds ratio of 3. Now, this odds ratio is not 10, but it's 3. This odds ratio isolates specific intrauterine effects because it controls to a large extent for the genetic predisposition for type 2 diabetes inherited from mothers by offspring. And it also controls for shared familial environmental exposures because these are siblings that live in the same households. On the right side of the slide, the same association is not found among sib pairs born before or after their fathers were diagnosed with type 2 diabetes, again suggested that these are true specific intrauterine effects and that this association is not just due to genetic factors or chance or birth order effects. So this is a powerful study. It doesn't tell us what the specific intrauterine effects are, but it tells us that they exist. And finally, just to put this into the public health perspective, why do we care about this strong association that is probably reflecting specific intrauterine effects? Likely reflecting specific intrauterine effects? Well, we care about it because of its public health impact. So what you see here is the population attributable risk for youth onset type 2 diabetes conferred by these intrauterine exposures. Diabetes exposure in utero, obesity exposure in utero, or both. So in the search case control study shown here, we created mutually exclusive exposure categories, and then we calculated population attributable fractions for type 2 diabetes in youth attributable to each of these mutually exclusive exposure categories. And as you can see, almost 47.2%, almost 50% of type 2 diabetes in youth at a young age can be attributed to these intrauterine exposures, which also means that could be prevented if we knew how to treat or prevent these conditions during pregnancy. However, as I mentioned, although we have very strong evidence with respect to pregnancy diabetes, it's not just pregnancy diabetes that programs the risk for chronic diseases down the road in the offspring. So we have, and everybody's now moving beyond just maternal diabetes, looking at a multitude of other risk factors and exposures. And here I'm going to start talking about our Healthy Start cohort study, a study that we started in 2009 by enrolling a pre-birth cohort of a little over 1,400 mothers from Colorado who were followed from early pregnancy through delivery and now into childhood via our participation in ECHO. At the beginning, we were primarily focused on maternal conditions during pregnancy, maternal obesity, diet, physical activity, diabetes, of course. But as we developed the cohort, we started to also think about other intrauterine exposures, maternal depression, exposure to environmental chemicals. With funding from ECHO, we were able to add a focus on the above the skin environment, air pollution, built environment. We also designed the cohort to very carefully look at biological signatures of these exposures in maternal samples, in cord blood samples, in offspring samples down the road. And as main outcomes, of course, we initially were focused a lot on offspring adiposity and growth, cardiovascular and metabolic outcomes. But then with the advent of echo, we have added outcomes related to brain development, neurocognitive outcomes, additional behavioral outcomes, respiratory outcomes as well. And so Healthy Start is now part of the ECHO consortium, and we are keenly studying the integration of the early life exposome outcome relationships and studying potential mediators and mechanisms responsible for associations that we describe. One of the things that from the start we were very focused on, given our research interests, was the assessment of offspring adiposity in our cohort. So we measured very carefully a lot of adiposity markers and markers of child growth at birth, at 4 to 6 months of age, at 4 to 6 years of age, and now funded by ECHO at 8 to 10 years of age. We not only measured weight and height or length, skin fold and circumferences at each of these time points, as well as supplemented with data from medical records, but we also measured body composition via method based on air displacement plethysmography that helps us measure fat mass, fat-free mass, and fat mass percent. And what you're seeing here on the slide is for the infants, how we measured that with the P-pod, and for the older children with the bod-pod, with pediatric option. The bod-pod device actually can be used for adults as well. And on top of this, we're also including measurements of abdominal fat and fat deposition via MRI to quantify the visceral, subcutaneous fat, and also measures at fatty liver. And we have these measures collected at 4 to 6, and we're collecting them now at 8 to 10 via our participation in ECHO. And I think it is important that we focused on measuring the adiposity so well. I'm going to try to make these points throughout my presentation. So what you see here is some information about how neonatal adiposity predicts child obesity. So until very recently, our early life marker of future child obesity risk was solely birth weight. And birth weight is a marker of future obesity risk in the offspring, but it's a very crude marker. And we know that for the same birth weight, 2 infants might have a very different body composition, as shown here. So we are now able to look at how adiposity tracks from birth to 5 months of age, 5 years of age, and going forward. And we found that for each one standard deviation increment in neonatal adiposity, we're seeing 0.12 units higher BMI and 20% higher prevalence of overweight and obesity in children between ages of 2 to 6. So these are novel data that show how adiposity tracks from birth onwards, and they also show that a higher percent fat mass at birth is associated with a higher prevalence of childhood obesity. And the reason why this is important is because it gives us another marker of growth and body composition at birth, in addition to birth weight, that is associated with future risk of childhood obesity. And it is perhaps a better marker of specific intrauterine exposures that might affect particularly or only adiposity and not birth weight. So it's a more sensitive marker, more specific marker of intrauterine exposures that increase the risk of childhood adiposity down the road. I'm going to show you some data from the Healthy Start cohort and some potential future opportunities for us in ECHO if we are to kind of replicate or leverage some of the findings from our cohort. So we studied a lot the prenatal metabolic environment because we're interested in maternal diabetes, maternal obesity, and associated conditions. So in one of our papers, we saw that, or we found that higher maternal pre-pregnancy BMI, but also higher or increased maternal gestational weight gain were independent of each other, associated with increased adiposity, percent body fat, at birth as our specific marker of interest. So the fact that these were independent association was important. And the other important thing was that the association between increased gestational weight gain and infant neonatal body fat was seen in all 3 trimesters of pregnancy. That is also during the first trimester of pregnancy, when this association between maternal gestational weight gain and in neonatal adiposity is not confounded by fetal growth, like in the, say, 3rd trimester of pregnancy. So these findings support a hypothesis that greater maternal weight gain during pregnancy, regardless of pre-pregnancy BMI, is directly related to offspring adiposity and at birth, and gives us support for interventions during pregnancy that focus on maternal weight gain to prevent offspring adiposity down the road. Now, of course, one would ask the question, so what is it about maternal obesity being a risk factor for childhood obesity? Same as we did with maternal diabetes and offspring type 2 diabetes. Of course, women with overweight or obesity during pregnancy have elevated maternal fuels, as shown here. Maternal glucose, maternal triglyceride levels, free fatty acids, and HOMA estimated insulin resistance are all higher in overweight and obese women compared to normal weight women, as shown here. And these associations exist for these fuels throughout their entire distribution. So there are no inflection points here necessarily. The higher the glucose levels, the higher the maternal BMI, the higher the glucose levels are, even within the normal glucose ranges. And all these fuels, glucose, triglycerides, free fatty acids, cross the placenta. So they are nutrients for the fetus. And this is what we call here the fetal overnutrition hypothesis, because since they cross freely the placenta, they're gonna overnourish the fetus even before the baby is born. So the question here is, which of these fuels then are responsible for the strong association between maternal BMI and neonatal adiposity? So we did here a structural mediating equation analysis in which we developed this DAG to test which of the fuels really mediate the association between maternal BMI and neonatal adiposity. So starting with maternal pre-pregnant BMI, going through maternal estimated insulin resistance, glucose, triglyceride, free fatty acid levels, and neonatal fat mass. And really the only pathway that was significant and mediated 20% of the association between maternal pre-pregnant BMI and neonatal fat mass was a pathway that included maternal insulin resistance and glucose. So glucose levels, even within the normal range, mediate 20% of the association between maternal pre-pregnant BMI and neonatal adiposity. And this pathway was significant. None of the others significantly mediated portions of this association, which contradicts some other reports that found that maternal triglycerides or free fatty acids are important mediators. But to our extent, we are the only study that looked at this role of maternal fuels as mediators in a general population cohort, not in high-risk groups, such as only in obese mothers, for example. So it's interesting that we're seeing again maternal glucose as being one of the fuels of interest. we are certainly interested in the association between maternal glucose or markers of glycemia and type 2 diabetes or offspring glycemia. So what we looked at here was, is there really an association between maternal and offspring glycemia? So not just maternal type 2 diabetes and offspring type 2 diabetes, but maternal and offspring glycemia continuously and in the normal range. And we've seen that children of women in the highest versus the lowest tertile of hemoglobin A1C had higher fasting glucoses at ages 4 to 6 or 4 to 7. Perhaps not an unexpected association, but certainly one of interest. And then we asked the question, is this associated then mediated by childhood adiposity? Because one would expect that childhood obesity or adiposity is the link between maternal and offspring glycemia. And to our surprise, we did not find significant mediation effect of this association by child adiposity, suggesting that although maternal glycemia, maternal diabetes programs or increases the risk of childhood obesity, and it also programs or increases the risk of childhood type 2 diabetes and childhood glycemia, there are additional mechanisms that do not necessarily or only go through childhood obesity that predispose offspring of women with elevated glucose levels to develop perhaps diabetes in the future. So more work, we're interested in exploring these mechanisms more in the future. Another effect of maternal obesity that we've been able to explore in our cohort once we started to add brain MRIs to our middle childhood participants, we found that maternal obesity alters offspring brain function in middle childhood. And in what way? We found here, panel A on this figure, a greater neuronal activity in major regions of the default mode network, the light gold highlighted areas shown here in children offspring of mothers with obesity compared to those of mothers without obesity. And these findings are consistent with findings in adults already having obesity. We also found subdued neuronal activity in a major region of the salience network, which may indicate altered processing of salient and rewarding stimuli. So perhaps the predisposition of for obesity in offspring of overweight obese mothers has to do with some of these structural brain changes that we detect even in normal weight young children. So lots to think about here and potential collaborations within ECHO to leverage these observations and add additional insight. We studied also in many of our publications the prenatal lifestyle environment. For example, we found that increased physical activity, but only during late pregnancy, is associated with less infant adiposity. We measured physical activity in our mothers 3 times during pregnancy, so we were able to do trimester-specific assessments. So we found that neonates of mothers in the highest compared to the lowest quartile of total energy expenditure during late pregnancy had 41 less grams of less neonatal fat compared to those in the highest quartile of total energy expenditure. And of interest, this association was specific to neonatal fat mass. There was no association between physical activity during pregnancy and birth weight or fat-free mass. So a very specific effect of this exposure on neonatal adiposity. suggesting avenues perhaps for interventions during pregnancy that would not affect birth weight or not increase the risk of small for gestational weight gain, but would have specific effects on adiposity. Similarly, we found that increased maternal energy intake from most nutrients, macronutrients, total fat, saturated fat, unsaturated fat, and carbohydrates were associated with increased neonatal adiposity at birth, independent of maternal BMI. There's no association of these nutrients with total fat mass, birth weight, and fat-free mass. So again, specific effects of these macronutrients on neonatal adiposity. And the other point that I want to make here is that it doesn't seem that one nutrient or macronutrient in particular has these effects, but it is more about maternal energy intake regardless of the composition of the macronutrients. Similarly, we did not find specific effects of some of the micronutrients that we looked at. If we looked at maternal diet quality using healthy eating index, we observed the same thing, that maternal diet quality is associated with, or lower maternal diet quality is associated with increased infant adiposity independent of maternal BMI and independent of total energy intake. And these effects are specific for neonatal adiposity. There was no association with fat-free mass or birth weight. The other lifestyle factor that we explored, smoking during pregnancy. There's a body of literature here about smoking during pregnancy and birth weight or postnatal growth. What's new for in our study is that we found that exposure to maternal prenatals or to prenatal smoking was significantly associated with reduced neonatal fat mass and fat-free mass. So growth restrictions in all body composition compartments, but as early as at 5 months of age, so not at 5 years of age, as early as at 5 months of age, exposed offspring had greater fat mass and fat-free mass than unexposed offspring after adjustment for birth weight. So exposure to prenatal smoking results in a growth-restricted phenotype that, and these babies exhibited very rapid early postnatal growth. And there is evidence that a rapid early postnatal growth increases the risk of obesity down the road. So this is an interesting pattern with growth restriction at birth and rapid growth early after that we're seeing with exposure to this toxic in smoking. And I'm going to highlight that we're seeing the same thing with exposure to some of the other chemicals that we're measuring via echo. As I was saying, with echo funding, we were able to focus one of our echo site-specific aims on better understanding these programming mechanisms. And I'm just going to give you a flavor of what we do. I'm certainly not the expert here. We're collaborating with basic scientists for this aim. But bottom line is that we're trying to measure and explore molecular signatures associated with in utero, metabolic, lifestyle, environmental exposures to determine biological pathways linking these exposures with child outcomes. And we're using 3 types of biological samples: placentas, infant mesenchymal stem cells, abbreviated MSCs here, and umbilical cord blood. We have more samples with umbilical cord. Almost 2/3 of our cohort has these samples available, and then placentas and MSCs in a smaller subset. But we have a substantial number of individuals with all 3 types of samples where we can interrogate some of these potential mechanisms. So here's what we're doing with these samples. So just a flavor of our placenta studies. They are conducted in Thomas Jensen's lab in collaboration with his team. So we have placentas from normal weight, overweight, and obese women. One of the studies here explored the hypothesis that from a diverse sub-cohort of healthy women across the BMI spectrum, placental proteins will be linked to child cardiometabolic traits, child glucose, triglycerides, adiposity traits, etc. Indeed, we found that proteins involved in the mTOR and AMP kinase pathway, inflammatory pathways, and insulin signaling pathways are indeed linked to child cardiometabolic traits as early as 4 to 6 years of age, as shown in some of these figures. These are pretty novel findings, I'm understanding, very rare to have such associations with proteins in placenta and traits down the road in the offspring. So one of our next steps in these studies will be to explore now which exposures, for example, does maternal quality affect these placental proteins to determine now how they mediate associations between exposures and outcomes. The MSC studies, I'm understanding also that this is quite a unique resource that we've created. The Kristen Boyle Lab is here, the pioneer. Even before ECHO funding, we were collecting these, we were collecting fresh umbilical cord tissue at birth, so not cord blood, but tissue from which MSCs can be isolated, grown, and stored, which we did. And we have 165 such MSCs that can be used for experiments. They're also in the freezer and can be used by other investigators. These MSCs are important because they can differentiate into all types of cells in the adult body, adipocytes, osteocytes, myocytes, chondrocytes, astrocytes. So they can be manipulated in vitro to go in one direction or another and study mechanisms. So one of the things we found was greater adipogenesis in MSCs from obese versus normal weight mother. They go the adipogenesis direction rather than the myogenesis direction, for example, mediated by the beta-catenin system. We also saw that DNA methylation in these MSCs correlates with percent fat mass at birth. And our most recent study is shown here. What directions do MSCs go when they are differentiated into adipocyte? Do they go the direction of adipocyte hypertrophy or hyperplasia? So the hypothesis here was that maternal obesity primes MSCs for adipocyte hypertrophy. And indeed, that's what we observed. We grew the MSCs from umbilical cord, measured proteins and gene expression, embedded these MSCs in hydrogels, induced adipogenesis, and after 14 days, they were visualized with a confocal microscope. So this study is showing that maternal obesity is associated with MSCs that overexpress adipogenesis proteins, such as ZFP423. I don't know a lot about this specific protein, other than that it's associated with the expression of over 500 genes enriched for cell cycle pathways, and then they differentiate into larger adipocytes. And I'm going to show you a nice picture about that. So the group's next step is to find out how does this model respond if the MSC hydrogels are embedded in mouse fat tissue. And these are the MSCs after 2 weeks of adipogenesis. The obese MSCs are 73% larger and store more fat, shown here in yellow, than the normal weight MSCs, confirming our hypothesis. We're studying also the above the skin environment. As I mentioned, this was an aim that has been facilitated by ECHO funding. And these are some of the directions that we're taking. We're doing air pollution studies in collaboration with colleagues from Colorado State University, Cheryl Maximin's team, studying traffic-related air pollution using GIS data and regulatory monitors, and black carbon. We've developed a black carbon spatiotemporal model, including new monitoring at residences. Now this model can be used for observational epidemiologic analysis by our team, as well as the rest of the ECHO group if there is interest. For green space, we're collaborating with CU Boulder, Department of Geography. We're furthering our observations about tobacco smoke exposure through collaboration with Brianna Moore, who was our postdoc. She got a K99R00 on this topic and took it with her to Texas, but she's still working with us. Measuring cotinine in maternal urine during pregnancy and biomarkers of childhood secondhand smoke exposure in urine. And of course, with Ann Starling, which is one of our ECHO investigators in Colorado, we're studying chemical exposures, maternal serum PFAS and urinary phthalates, phenols, flame retardants, metals, and others. Some of the findings about environmental exposures and neonatal adiposity are shown here. We found an increased mid-pregnancy urinary, we found that increased mid-pregnancy urinary PFAS exposure was associated with lower adiposity at birth and lower birth weight, but higher adiposity at 5 months, especially among males, and rapid growth in weight for age up to age 5 months. Remember, this was also seen with prenatal smoking exposure, so a very similar pattern of growth in response to maternal urinary PFAS exposure. Also found that third trimester ozone exposure was associated with greater adiposity at 5 months of age. And now there are many other studies on these environmental exposures coming from our group, and Starling's leadership here is important. For example, distribution and predictors of urinary concentrations of phthalate metabolites and phenols in pregnant women in Healthy Start. So why are we seeing what we're seeing in terms of these exposures? The spatiotemporal prediction model for black carbon in Denver, the one that I just told you about, that it's available for our investigators and for others in ECHO to further explore. We're also interested in mixtures or combinations of exposures in early life. For example, the interaction between secondhand smoke, exclusive breastfeeding, and infant outcomes in our cohort, and also combined environmental and social exposures during pregnancy and their association with neonatal outcomes. This particular paper was first published for our cohort, but there is a manuscript proposal in ECHO, just kind of sitting there for 2 years to do something similar across multiple cohorts in ECHO. And even more recently, with the addition of Wei Peng to our group, we're starting to focus more and more on metabolomics across the life course. Of course, metabolomics can be used to better capture exposure, to refine outcomes, and or to understand pathways. And Wei is primarily focusing on metabolomics in samples during pregnancy, plasma, serum, urine, maybe others, to really look at how metabolomics mediates associations between exposures and childhood outcomes. She has an approved paper proposal in ECHO to do so in several cohorts in ECHO, and create some resources for future studies. I'm going to move fast to the role of postnatal lifestyle, because as I said in the beginning, these sensitive periods that we're exploring for developmental determinants of childhood obesity do not stop at birth or with early life, but continue throughout the life course. So we're interested in how we can use this postnatal period to kind of mitigate, in fact, the increased risk for obesity and metabolic outcomes in offspring exposed in utero to some of these harmful factors. So an observation from another cohort in Colorado, not the Healthy Start study, another study called EPOCH, that we believe is important. We found that breastfeeding modifies the effect of exposure to diabetes in utero on childhood BMI, childhood obesity. So what you're seeing here is body mass index in 10-year-old offspring exposed in red and not exposed in yellow to maternal diabetes in utero. And in those who were breastfed for less than 6 breast milk months, there's a strong association between exposure in utero to diabetes and higher BMI. Whereas in those who were breastfed for more than 6 breast milk months, this association is much reduced and not statistically significant. And the same was true for other markers of childhood adiposity, visceral adipose tissue, subcutaneous adipose tissue, and waist circumference. So this is a nice example of biological interaction between exposure during a sensitive period, diabetes in utero, and another exposure in postnatal life, breastfeeding, showing also that one can do something about mitigating, reducing the increased risk conferred by in utero exposure in terms of obesity prevention. Similar findings also in EPOCH with current childhood diet, estimated here based on healthy eating index. So in those who had currently a low healthy eating index, exposure to diabetes in utero was associated with much higher BMI, waist height ratio, visceral and subcutaneous adipose tissue. But in those with the current better healthy eating index, these associations were not significant. But as we know, most US youth do not meet the recommendations for eating healthy, exercising. What can we do? Maybe not much, maybe a lot, but we have to do something. We think that we have to take a comprehensive approach to prevention throughout the life course. And just to put this in context, I'm showing you here what happened with diagnosed diabetes in adults in the United States. After a steady period of rising rates, we're now seeing perhaps that we might have turned a corner where might be seeing for the first time in 20 years an annual rate of diabetes development in adults that is decreasing. That's certainly good news, and maybe that's the consequence of the landmark diabetes prevention program and its implementation as the national DPP, which is now reimbursable with Medicaid and Medicare. So great news. So this might be going down at a rate of 5.4% per year. But remember what I showed you in the beginning of this talk? The rate at which type 2 diabetes is increasing in children? 4.8% per year. So almost the same relative change, but in the opposite direction in children compared to adults. So this tells us that this is not enough. It's great news, but it's not enough, because what we are seeing now is arise in chronic diseases like diabetes much earlier in life. And we have to do something about it. We have to continue our focus on high-risk adults, but also think about primordial prevention in children. And here I'm just showing an example of a study that we are doing, primordial prevention in high-risk American Indian youth, Tribal Turning Point. It's a DPP-based intervention for American children aged 7 to 10 years. Has a curriculum, has individual MI sessions and a toolbox. So these are data from the pilot phase of the study, in which we studied 62 youth in Navajo and Eastern Band Cherokee. And although the study was a pilot, it was not designed or powered to see significant differences. We have seen significant treatment effects. Which is why we are now conducting the full-blown randomized clinical trial with American Indian children in southwestern American Indian tribes. And stay tuned for results. We're in the third year of administering the intervention and collecting the data. And finally, another future focus for us, and maybe for ECHO as well, we have strong evidence from observational studies such as Healthy Start, EPOC that I talked about, that pre-birth exposures influence obesity and diabetes risk across the life course. But we also have close to 100 studies conducted during pregnancy, prenatal interventions conducted during pregnancy that have shown little effect on maternal gestational weight gain, maternal diabetes, or birth weight in terms of offspring outcomes. Now, of course, birth weight, as I showed you, is not the best outcome, but so many interventions that showed a little effect. So we believe that earlier intervention is needed, and we need to focus now preconceptionally. So we're moving our efforts to promote preconception health, and this is work that is conducted by Kate Saller. I can't tell you much more about this because her grant is under review right now. We hope it's going to be funded, and then we'll have more exciting news to discuss with you. So future research. I am of the opinion that we need to continue the systematic and sustainable surveillance of pediatric obesity and diabetes. This is something that we often forget about, that in order to know what's going on, we need surveillance systems. And some are better than others, but we need to do this and expand upon this to better understand the burden and risks, the effects of prevention programs, development and burden of complications and mortality. We need longitudinal pre-birth cohort studies and consortia of such studies like ECHO to comprehensively explore the role of environmental exposures and their biological signatures and pathways, and to identify potential causal associations. We need improved understanding of mechanisms, such as mechanistic studies nested in population cohorts like we are doing with our programming and basic science components of our ECHO research. And finally, we need effective interventions, randomized clinical trials, and translation studies, but we definitely need to increase the focus on primordial prevention in youth, and to switch a little bit into the preconceptual health, as difficult as it might be. And I want to highlight here the LEAD Center, which I have the honor to be directing. We're trying to increase the diversity of our staff and colleagues, and now I think we have 2 gentlemen instead of 1 who are part of the LEAD Center, and we're going to do our best to continue to increase that diversity. Thank you.